Hi, everyone. Sorry about that little glitch, but that's kind of the beauty, I guess, of technology. I can just finish recording it. So we had one slide left on that last lecture. I'm going to add it to the cardiovascular 3. I'm in the process of uploading all the today's lecture on YouTube, so I will be sending that out to you as soon as possible. So just to finish up with the cardiovascular 2 lecture, where we left off was right here. And we were talking about, the mechanism of contraction. And remember, of course, cardiac muscle, or otherwise known as EC coupling, excitation contraction coupling, involves an influx of calcium from the outside, which activates the release of calcium from this SR, what we call calcium-induced calcium release. Calcium then leaves, binds to troponin, triggers the contraction. And then, of course, to end the contraction, calcium is pumped back both into the SR and also out through that sodium-calcium exchange. All right, so I'm going to continue now with the next lecture, which is cardiovascular 3. Let's see. Yeah, so just as a reminder, I'm not sure if I had it on the last slide. Calcium comes in with heart muscle, calcium comes in from the outside through calcium channels, binds to the ryanodine receptor, triggers the release of more calcium. We call it calcium-induced calcium release. This whole thing is called EC coupling, excitation, followed by contraction, coupling. Calcium is released, binds to troponin, triggers the sliding filament. Relaxation occurs by calcium being pumped back both into the SR, where part of its stores came from, and also out of the cell via the sodium-calcium exchange. All right, so I will now continue with the cardiovascular 3 lecture. If you just give me a moment here. Let's see. Share screen, and here we go. All right, so the third cardiovascular lecture is on blood. And I'm not going through in a lot of detail, probably the least of all the different parts of the cardiovascular system, but definitely want you to be familiar with it. So far, the focus has been on the heart and properties of the heart in terms of how blood is pumped through the heart itself, the valves, diastole, systole, factors that regulate stroke volume, all those different factors, autonomic effects, venous return. Today we're going to focus on blood, and then on Thursday we'll focus on blood vessels themselves, the third component of the cardiovascular system. And that will lead us into the respiratory system. All right, so let's see, here we go. All right. All right. So the blood. So what can we say about the blood as far as what is found in the blood, right? This is an important vehicle that carries hormones, it carries nutrients, removes waste, a lot of different things, but also it consists of other important cellular components. So all I want to focus on at this point is recognizing the fact that whole blood is divided really up into two main parts, what we call the plasma, which is the liquid portion, kind of a straw colored, And then what we call the packed cell volume, or basically really with mostly the red blood cells. The plasma makes up about 58%, roughly, can be more or less. And then the blood cell volume, which is mostly red blood cells, about 42%, maybe 1% white blood cells, make up the rest. Red blood cells are also known as erythrocytes, and white blood cells are known as, you can see there's 5 different classes of white blood cells, and we'll talk about those. And we'll also talk about platelets, that we call the cellular elements of the blood. All right. So to kind of summarize, but from the last slide, whole blood is divided up between plasma, which anywhere 55 to 58%, which is primarily composed of plasma proteins, some solutes, as well as formed elements such as blood or inorganic molecules, organic molecules, which you can see are divided up into these different categories. The plasma proteins consist primarily of albumin, which has a very important role in terms of osmoregulation. Some other albumins, as well as globulins, are important in transport. They're kind of like a nice little shuttle system. Fibrinogen plays a very important very important role in the clotting mechanism, which we'll talk about on Thursday. And then we've got a few other regulatory proteins, such as enzymes. We also have a lot of ions, as you can see, sodium, potassium, a lot of the ones that you're familiar with, as well as nutrients, as well as waste material. So that's really what the plasma is made up of. I mentioned that albumin plays a very important role in osmoregulation in terms of controlling movement of material into and out of the blood. But I also wanted to revisit a little bit about some of the hormones that play a role in osmoregulation. And really, osmoregulation is a way, if you remember, of maintaining the salt and fluid balance of the blood, which is very important. There's 4 main factors that regulate plasma volume to making sure we have an adequate amount of plasma in the blood. One is our ability to respond to the thirst sensation. When we're dehydrated, the hypothalamus is activated via its osmoreceptors, which gives us the sensation or the need to drink. And of course, when you drink, that will increase your fluid volume, which ultimately will increase your plasma volume. There's 3 other hormones that also that are involved in osmoregulation. One is ADH, or vasopressin. We discussed that earlier. This is a hormone that's produced when we're in a dehydrated state, or basically low fluid volume. And what this does, it helps to kind of increase the fluid uptake. We also have what's known as the renin-angiotensin-aldosterone system, which is something we'll talk a bit more when we focus on the kidney. This is a system involving enzymes and hormones, primarily aldosterone, that responds to low sodium. So the response to low sodium is to uptake more sodium into the blood. With ADH, the response to dehydration is to bring up more water into the blood. Finally, we have ANP, or ANF, atrial natriuretic peptide or factor. This is a hormone produced by the blood. By the heart. I had too much caffeine this morning. By the heart, and this responds to with the blood pressure or blood volume is too high. This results in excretion of fluid as well as salt. Remember, ADH is produced by the hypothalamus, and aldosterone is produced by the adrenal gland. And this is kind of a neat little picture as well. Here we've got from the hypothalamus via the pituitary, we have vasopressin that promotes the uptake of water. And then aldosterone from the adrenal gland promotes the uptake of sodium. Both of these in response to low levels of each. All right, so that's all I want to say about plasma for now. I want to really zero in on the cellular elements. Why don't we just call them cells? Well, because we do have some cell fragments that play a very important role. So they're not true cells, but they are fragments. Those, in fact, are platelets. All right, so the cellular elements consist of erythrocytes, which are the red blood cells, which make up about 99% of the packed cell volume. We have leukocytes, which are white blood cells and are divided up into 5 different types, where we'll just kind of scratch the surface as far as discussing them today. And then those, of course, are involved in protection, part of the immune system, defense. And then finally, we have platelets, which play a role in clotting. All right, what can we say about red blood cells, first of all? Well, first of all, when you look at a red blood cell, they don't look like a cell at all. I mean, they are round, but their shape is like a biconcave disc. They basically lack a lot of the things that most cells do. They do not have a nucleus. They do not have any organelles. There is cytoplasm, of course. The main volume is comprised of hemoglobin. So basically, the red blood cell is a sack full of hemoglobin. But that hemoglobin, of course, is very, very important because it's that hemoglobin that's involved in transporting oxygen and, to a lesser extent, CO2. This biconcave shape and the lack of organelles helps to increase the surface area of the red blood cell, which will maximize the amount of oxygen it can carry. All cells have a lifespan. Red blood cells, typical lifespan is about 120 days. Under certain pathological conditions, such as anemia and things like that, it can be less. So what happens at 120 days? Well, red blood cells are typically starting to wear down anyway, showing signs of age. And what happens is they are removed from the circulation by the liver, bone marrow, as well as the spleen. These tissues have the ability to recognize when red blood cells are getting old. They have changes in their membrane that are a flag, an indicator that they're getting old and fragile. When red blood cells with their hemoglobin are broken down, one of the products that's formed is bilirubin. And those of you that work in the clinical setting, you're familiar with bilirubin, measuring blood levels of bilirubin, which really should be very low, if at all present. That's because bilirubin is typically excreted. The only times where you will see bilirubin in the blood is in cases of jaundice. Someone has liver issues where there's a buildup of bilirubin in the blood and it's not excreted. And also, in a normal sense, many infants at birth have jaundice. Their skin is kind of a yellowish color. That's because they do not yet have the ability to metabolize bilirubin. So you see levels elevated. This typically can be eliminated by ultraviolet exposure, which is typically done in a clinic or a hospital. All right, little chemistry with hemoglobin. What is hemoglobin? Well, hemoglobin, you can see right here, is a collection of 4 polypeptide chains. 1, 2, 3, 4. They're called globin chains. We have 2 beta chains, these 2, and we have 2 alpha chains, and that has to do with their structural features. So 2 beta and 2 alpha, and you can see 2 beta, 2 alpha. Within each of these 4, we have a molecule in the center known as heme. And heme is the iron-containing compound, which is very important because it's the iron, it's the heme where the oxygen is going to bind. So you wonder, where on this molecule do oxygen and CO2 bind to? They bind right to there, that little heme molecule. Like I said, red blood cells are removed by phagocytosis via the liver, spleen, and bone marrow. And what's interesting is this last part. The hemoglobin, which is broken down to bilirubin, that doesn't necessarily all go to waste. Some of it is excreted, but we'll learn a lot of the components of the red blood cell are recycled. It's a very extensive process, but very important. I've got a diagram here you can see and the description. It's kind of a busy diagram, so just to kind of orient you, there's the bone marrow. This is meant to represent macrophages in the bone marrow, but primarily in the spleen and the liver. Here's the organ itself, and here's the kidneys. So if you want to trace the path of how red blood cells meet their fate, red blood cells are first formed in the bone marrow, right? Some die off initially, but most of them, they're released into the circulation. And by about 120 days, like I mentioned, the membranes of the red blood cells change. They're modified, and they're looking old and ragged, so they're removed. And they're removed by macrophages, excuse me. Thank you. By 120 days, they're removed. And of course, the main component of a red blood cell is what? Hemoglobin. So what happens to the hemoglobin? Well, hemoglobin, remember, consists of globin and heme. And heme contains iron. So first thing that happens when the red blood cell is phagocytized, the globin portion, which is the protein, is degraded into amino acids and recycled back to the bone marrow, where it can be reused. Next, we have the heme. Now the heme, there's the heme molecule, and then we got the iron. The iron is also recycled by way of a blood protein called transferrin, which shuttles it also back to the bone marrow where it's recycled. That leaves the heme without the iron. And it's the heme that's first degraded into a molecule called biliverdin, ultimately into bilirubin, which ultimately has a couple different fates. The bilirubin is incorporated into a substance called bile, which I know many of you guys have heard of. Bile is a very important molecule in digestion, which helps to break down fats, and you can see most of it ends up being eliminated. So most of the bilirubin by way of the liver is eliminated. Some also travels to the kidney, where it is also eliminated. So some of it is recycled, but most of it is eliminated. Excuse me for my stuffy nose. All right. From the red blood cells, let's go to the white blood cells. And white blood cells are also known as leukocytes. Leuko means white. Several different types. I'm not going to get into all the details of every single type of white blood cell, but they're characterized by having this ability to move like an amoeba, amoeboid movement, in the sense that even though, yes, they're found in the blood, they can squeeze through spaces in the capillary and enter into tissue where they can encounter a pathogen or some sort of injury that they can deal with. So kind of like a first responder type thing. It's called diapedesis. White blood cells are divided up into two types, what we call granular leukocytes, which are the neutrophils, eosinophils, and basophils. Then they're characterized by the staining of these granules. Eosinophils, the granules stain with a eosin or an acidic stain, basophils with a basic stain, and neutrophils more neutral. These are typically phagocytic, produce a lot of enzymes. Then we have the agranular ones, which are ones that don't have any observable granules. Now, there are granules there, as you can see. I mean, all cells have granules, whether they're vesicles or whatever, but you can't see them with these cells. They're very small, whereas these, under the microscope, they're very clear, as those of you guys that have taken anatomy or looked on Google Images. These include lymphocytes, which produce antibodies as well as other things, and monocytes, which are phagocytic. These are the largest. The largest white blood cells are the monocytes. Like I said, it's going to be very brief, our discussion with that. What about platelets? So platelets are very interesting, kind of one of my favorite, if I had to pick one, of all the different cellular elements. They're also known as thrombocytes. They're the smallest, and they're actually a cell fragment. So you wonder, why are we talking about a cell fragment? Is it doing anything? Well, in fact, it's a fragment of a much larger cell called a megakaryocyte. This is a huge cell that normally is only found in the bone marrow. And during the process of development, there's little bits of the megakaryocyte that are shed. See these little pieces that come off of it? These are the platelets. Now, what's so special about them? They are packets full of a variety of different enzymes that are important in the process of what we call hemostasis. Not homeostasis, but hemostasis, which is the process of blood clotting. They contain some, for example, serotonin, which you've already heard of in the nervous system, but serotonin with platelets is important for constricting blood flow, which also explains its role in treating people who have migraines, as far as regulating, preventing any vasodilation. It also produces a lot of other chemicals that maintain the integrity of a blood vessel, which of course is injured, right? Why do we need blood clotting to occur when a blood vessel is injured? These fragments have a very short lifespan. I didn't talk about the lifespan of white blood cells because it's extremely variable. Lymphocytes can stick around for years, it's many of them, whereas neutrophils and things like that may only last a week or two. So really variable. Okay, so we've talked about the different cellular elements, but let's talk about how they come from, where they're formed and how they become to be. So in everyone in the room, anyone actually postnatally, post-birth, the bone marrow is the organ where blood cells are formed, right? Before birth, during fetal development, the liver plays that role. So these are all the different blood cells that I mentioned. And you can see if we follow back, kind of going backwards, there's several different levels of cells where they come from if you follow the arrows. Ultimately, there's a stem cell. We'll take a look at in a second, way up here, that is kind of like the mother of all blood cells. And that differentiates, eventually forming the blood cells. How does a stem cell start to produce erythrocyte versus a white blood cell versus a platelet? Well, there happens to be certain chemical factors that are responsible for this, right? Now, there's chemicals are involved in everything that triggers stem cell development. If we divide hematopoiesis into three categories, erythropoiesis, which is the formation of specifically of red blood cells, that's dependent on a hormone called erythropoietin, which is a hormone that's produced in the kidney, plays a very important role normally in red blood cell formation, but it's also used clinically for people who are suffering from certain types of anemia. Then we have leukopoiesis, which, as you might expect, is the formation of white blood cells. And this depends on the production of what we call cytokines. CSF, that's not cerebrospinal fluid, that stands for colony stimulating factor. But there's 5 types of white blood cells, and they're dependent on different cytokines. For example, cytokines that trigger formation of neutrophils are different than the ones that lead to formation of lymphocytes. Finally, thrombopoiesis, which is the formation of platelets from, as you can see, this megakaryocyte. leads to is dependent on the formation or the hormone thrombopoietin. Finally, to look at the big picture, that stem cell is known as a hemocytoblast. And you can see it branches in all directions. Here we've got different CSFs for the different white cells. Here we've got erythropoietin for the red blood cell. Here we've got TPO or thrombopoietin for the platelets from the megakaryocyte, which is a very large cell, to form platelets. All right. So, are red blood cells formed all the time? Yes, they are, just like all blood cells are. Are there certain conditions that can increase or decrease the production? Well, there are conditions that will increase the production of red blood cell, and that has to do with the amount of oxygen. It kind of makes sense, right? If you're low on oxygen, you would assume that you'd want to make more red blood cells to help carry whatever's there. So in fact, if you look at this diagram right here on the right, here's the conditions, for example, low oxygen, high oxygen. If oxygen levels as detected by the kidney typically are low, this triggers the production of erythropoietin. Erythropoietin induces this differentiation and ultimately the formation of red blood cells. And of course, once you have more red blood cells, that's going to improve the transport of oxygen through the body. It's going to be a more efficient carrier. If we take a look at the development of a red blood cell over time, it's very interesting, right? Here's our hemocytoblast, the stem cell, and you can follow along these steps all the way to a red blood cell. Now, I don't need you to know all of these steps, but I do want you to notice a couple key things. First is at what we call the normoblast stage. It's at the normoblast stage where we're starting to see a cell which has a nucleus in it reach the point where that nucleus is ejected. All right, nucleus is ejected. And then it becomes a reticulocyte. That reticulocyte, once it starts to make hemoglobin, becomes an erythrocyte. Hence, you can see the red. All right. Okay, so the last bit I want to get into is the concept of blood typing. And many of you guys know your blood type. Some of you guys don't. Normally in lab, we would do this exercise. Unfortunately, we won't be doing it unless you want to stick yourself with a little pin and collect a blood sample, which you probably don't. What determines the blood type has to do with the presence of a protein on the red blood cell that's collectively known as an agglutinogen. Now, these agglutinogens, you're probably familiar more with the name of like A and B, right? So if someone has an A agglutinogen on their red blood cell, they have type A. If they have the B agglutinogen on their red blood cell, they're type B. If they have both, they're AB. If they have none, they're type O. There's another factor, another protein called the Rh factor, which came about because it was discovered first in the rhesus monkey. If you have that Rh factor, you're positive. If you don't have it, you're negative. Now, normally, unless someone has a very serious autoimmune condition, your immune system sees these proteins and ignores them. It's like, okay, this belongs to me. But of course, you've heard of people who get transfusions of the wrong blood type, right? And if it's not a match, that can cause a problem, very serious, potentially fatal problem. And this has to do with the presence of antibodies. We all have them in our plasma, right? So we've got these proteins or antigens on the surface of our red blood cells. And in our plasma, we have antibodies that are targeted, just like antibodies are, in this case, targeted towards proteins that we don't have. So for example, if you have type A blood, you have antibodies that would attack type B. If you have type B blood, you have antibodies that would attack type A. This is a way of protecting you from an incompatible blood type. All right. So this is a diagram which illustrates several things. Number one, you can see the different antigens on these red blood cells. Almost looks like the spike proteins of the COVID. But anyway, you can see kind of the proportion throughout the population. This is the US population, roughly. I fit into this category right here. Most common, of course, is O. So this is the antigen. You have the corresponding, the antibody that you don't have against the protein you don't have. So for example, if you're type A, not personality, but blood type, you have anti-B antibodies. If you have type B blood, you have anti-A. If you are AB, you don't have any antibodies. If you don't have any, if you're type O, you have no antigens, but also both antibodies. So going a little bit further, this kind of gives you an indication, explains why we can, who we can receive blood from and who we can donate to. For example, if you're type A, you can receive blood from someone who has type A blood, and you can receive blood from someone who's O, who doesn't have any antigens. If you're type B blood, hey, David. Hi, David. Oh, well. And if you're AB, you can accept all the different types of blood.
I have a quick question, Kevin.
Yeah, hi, David.
Is this this isn't the 235 lab, is it?
No.
Okay.
No, this is the second. So I'm doing cardiovascular 2 and 3.
Okay.
I'm actually recording it. So even if no one's there, it's going to be recorded, but thanks for stopping in. Yeah, for some reason, like right towards the end of that first lecture, my internet got messed up.
Right.
No, this,
I've been having trouble with internet too.
Yeah, it's kind of a little unstable.
Yeah. Well, I'm still looking for my, you don't have the code that Robin uses for her lectures, do you? Do you know what that is? Because I can't find it in my inbox.
Ooh, did you try emailing her?
No, but I'll text my lab partners. I'm sure they'll have it.
Yeah, after I'm done here, I can go ahead and check for you.
Okay, thank you.
Yeah, sure.
Bye.
Bye-bye. So what else? Yes. So if you're type AB, you don't have any antibodies, so you can accept blood from everybody. You're considered to be the universal donor. Ultimately, it's true if you have the Rh factor. So if you're AB positive, you don't have any antibodies. You can accept blood from anybody. You can only donate to people that are AB. If we were all together, I can kind of explain it more. This is sort of just a kind of a quick and dirty chart. If you're type O, because you have antibodies to both A and B, you can't accept blood from anybody except O. But the cool thing is you're a universal donor. And when you donate, you're looking at the red blood cell, not the plasma. So someone who's type O, no antigens, they can donate to everybody because nothing for the person's antibodies to react with. But of course, you can only receive blood from O because you've got that. That's when you're looking at the antibodies. So when you donate, it's your red blood cell antigens. When you receive, it's the antibodies. All right. This is just a diagram showing, like I said, when considering whether a transfusion will be compatible, it's most important to consider the donor's antigens and the recipient's antibodies. And this is what happens when you have a transfusion reaction. Say, for example, you try to donate to someone who's type A who happens to have anti-B antibodies, you're going to get clumping. if you're type AB and you try donating to someone who's type B, has anti-A antibodies, it's going to clump as well. So, all right. There's a really neat website, and let me see if this is still good. It used to be. Yeah, I'll send the other one. There's a newer version of this that's a really neat, like, blood typing game. All right. All right, so the Rh factor I mentioned is another group of antigens besides the ABO story. Like I said, if you have the Rh factor, you're known as positive. So if you don't have any antigens, but you've got the Rh, you're O positive. If you're, for example, if you have the A antigen, but no Rh, you're A negative. So these are the different options, right, that you can have for blood type. Now, what's interesting is with the ABO story, whatever antigen you have, you're born with antibodies against what you don't have. For example, if you're you have A blood, you're going to already have anti-B antibodies. If you're B blood, you're already going to have anti-A antibodies. The difference with Rh is you only develop Rh antibodies if you're exposed to someone who has Rh blood type. This can happen through placental sensitization, which I'll talk about, or during blood transfusion. All right. So, formation of Rh antibodies. Like I mentioned, unlike people who are AB, we're looking at AB grouping, people who are Rh negative do not automatically have antibodies against Rh. They will only get antibodies, they will only form them once they're exposed. So hopefully you kind of get that. With the AB story, you already have antibodies against what antigen you don't have. These antibodies you will get during an incompatible transfusion. Say if a person who's A positive will donate to someone who's A negative, meaning this person has the Rh antigen, this person doesn't, and someone will develop antibodies to it. Or during pregnancy, if the mother is Rh negative and the baby she's carrying is Rh positive, right? Then the mother will develop antibodies to it. Now, this is a very interesting clinical situation because if you have that scenario where you have an Rh negative mother carrying an Rh positive baby, this can actually lead to a condition called hemolytic disease of the newborn, or otherwise known as erythroblastosis fetalis. Now, how does this occur? It will not happen during the first pregnancy. So if you take a look for an example, here's a couple. Say the eventual father is Rh positive. See all these little positive plus signs. Here, the potential mother is Rh negative. Just by the roll of the dice, whatever, this person's going to be carrying an Rh positive baby. What can happen is not during pregnancy, but at the time of childbirth, when all the membranes rupture, the Rh positive blood from the baby, right, when the membranes rupture, will come out and the mother will then be exposed to it. So it's only at childbirth when the membranes rupture, because normally there's a maternal barrier with the fetus. The two bloods don't see each other. But at pregnancy, that's when the Rh positive blood of the fetus can enter into the woman's bloodstream. The mother will develop antibodies. That won't be a problem for this baby that was just born, right? It's being born. But say that woman gets pregnant again and she carries a baby who's Rh positive. The antibodies that mother developed, what will happen will then go through the amniotic sac into the baby and attack the fetal red blood cells. All right. So normally in most countries, I don't know about all, there's a precaution that's taken. Any woman who's Rh negative, I should say, and is pregnant, routinely they're given a drug called Rogam, which will inactivate the Rh antigen on the fetal red blood cells so the mother does not see the Rh antigen. It's kind of a way of masking them, so the mother can't make antibodies against something that she doesn't see. All right, so that takes us to the end of that lecture. So that was cardiovascular 3. We finished cardiovascular 2. I'm going to go ahead and save this and send you guys the YouTube. And I'll see you on Thursday. If you have any questions, let me know. And good luck with lab. And